Welcome. Today we're going to shrink ourselves down, way down to the molecular level to get to know the most important and maybe the most amazing little machines in the entire universe, proteins. So to get started, let's just ask a simple question about a machine you know really, really well, your own body. What exactly is the engine that's powering all of this? You know, the answer isn't a single central engine like you'd find in a car. Nope. It's more like a massive, sprawling city with trillions of microscopic workers, and every single one of them is busy doing a very specific job.
And these tiny little engines are called proteins. Honestly, they're the foundation of what it means to be alive. They carry out almost every single function inside every living cell.
Okay, so let's dive into the story of how you build a protein. Like any good piece of machinery, it all starts with the blueprint.
So every single protein is what we call a polymer. Now, that's just a fancy word for a long chain made up of repeating units. And in this case, those units are called amino acids. They all have the same basic backbone, but see that R group? That's the special part, the side chain, that gives each one its unique identity.
And there are 20 of these standard amino acids, each with its own chemical personality. Some are what we call hydrophobic, which means they hate water, so they try to hide on the inside of the protein. Others are hydrophilic, they love water, and they're happy to hang out on the surface. And then you have others that carry electrical charges, kind of like tiny magnets, which are super important for a protein's shape and how it interacts with other molecules.
These amino acids link up together using super strong connections called peptide bonds, and that forms a long chain. The specific order they're in is what we call the primary structure, and you really can think of it like letters in a word. That sequence spells out everything, what the protein will become and exactly what it's going to do.
Now here's where it gets amazing. How does this simple, one-dimensional string of beads just spontaneously fold itself into a complex, functional, three-dimensional machine? Well, it happens in this really beautiful four-step process. So you start with the primary sequence. Then, that chain starts to form little local shapes, like tight coils we call alpha helices, or flat folded sections called beta sheets. Then the entire chain, coils, sheets, and all, scrunches up into a complex 3D shape. That's the tertiary structure. And for a lot of proteins, there's even one more step, where several of these folded chains get together to form an even bigger, more complex machine. So here's the absolute key takeaway. This whole complicated folding dance is completely programmed into that initial sequence. As the Nobel Prize winner Christian Anfinsen proved, the instructions for building this complex machine are written right into that chain of amino acids from the very beginning. Okay, so once our machine is folded, it's ready for its mission. And the sheer variety of jobs these proteins do, well, it's just staggering.
Seriously, proteins do pretty much everything in our cells. They're the enzymes that make chemical reactions happen at lightning speed. They're the structural beams that give our cells their shape. They're the delivery trucks that carry vital cargo around. They literally do it all.
Let's just look at a few examples. Myosin, that's the motor protein that powers every single one of your muscle contractions. And then you've got the builders, like collagen, this super strong rope-like protein that gives our skin its strength, and keratin, which is the tough stuff that makes up our hair and our nails. And this might be one of their coolest jobs. They're our personal bodyguards. Antibodies are these incredibly specific proteins that patrol our bodies. They're programmed to recognize and latch onto invaders like viruses and bacteria, tagging them so the rest of the immune system knows to destroy them. They are our front line of defense. But this incredible story also has a dark side, because sometimes that all-important folding process goes terribly wrong. Just look at the two different fates of a protein like this one, alpha-synuclein. When it folds correctly, it does its job in the brain, no problem. But if it misfolds, it gets sticky. It starts clumping together with other misfolded copies, forming these toxic clumps that actually kill nerve cells. And that is what leads to the devastating symptoms of Parkinson's disease. And this process, this misfolding and clumping, is actually the root cause of so many heartbreaking diseases. The infamous plaques and tangles in Alzheimer's, the Lewy bodies in Parkinson's, they're all caused by proteins that have just lost their way. It connects this tiny molecular story directly to our health.
But, and this is a really important but, here's the hopeful final chapter. By understanding proteins this deeply, we're now learning how to harness their power and even engineer them for our own good.
A fantastic example is how we make therapeutic antibodies. I mean, look at this timeline. Over the decades, we've gone from using mouse proteins that our bodies would often reject to engineering them to be more and more human. And today, we can create fully human antibodies that are some of our best weapons against cancer and autoimmune diseases.
And this kind of engineering is now hitting warp speed, completely transformed by the power of artificial intelligence.
That massive number you just saw, over 200 million, that's how many protein structures have been predicted by an AI tool called AlphaFold. In just a couple of years, it gave us the detailed 3D blueprints for pretty much every known protein. It's like we went from having just a few scattered maps to suddenly having a complete atlas for the machinery of all life on Earth.
And that leaves us with this really profound thought. For decades, science has been all about learning to read the book of life, our DNA. But now, by understanding and predicting the shapes of these protein machines, we are right on the edge of not just reading that book, but actually writing in it ourselves. So the real question is, what new chapters are we about to create?